Hello student, all of you are most welcome on my channel, that is Liberty Solution. In my previous video, I have discussed about chapter number 8, Human Health and Disease, class that is a portion of class 12th biology. And in that video, I have discussed about common diseases, what is health and common diseases in human being. In today's video, I will be discussing about immunity system, what are different types of immunity as well as allergy. If you have not subscribed my channel, subscribe it to get many more videos related with class 11th and class 12th biology. These videos are useful for your school examination, CBSE board examination, and in case if you are preparing for NEET examination, they will be very useful for you. So let's start today's lecture that is related with immunity, immune system, and allergy. So we'll be discussing about human immune system. This diagram shows human immune system. There are lymphoid organs are there, thymus gland is there, intestine is there. So I will be discussing out like there is a digestive system, there is a respiratory system. In same way, there is an immune system. What is the function of immune system? To develop immunity and help individual to fight against uh, attacks of pathogen or any antigen. So this is a tonsil. Tonsils are a part of what immune system. Lymph nodes. There are lymphatic vessels. Pierce patches are found in the intestinal wall. Red bone marrow, that is also a part of immune system. This entrance of thoracic duct into subclavian vein, there is also some part of immune system is there. Thymus is also a part of immune system. These are axillary lymph nodes. These are thoracic ducts and spleen and inguinal lymph nodes. So these are different locations where our immune system is present. So what is the human immune system? It is the system that gives immunity to the body by recognizing, responding, and remembering foreign antigen. Foreign antigen, it may be any bacteria, any virus, any fungus, any chemical. So that will be foreign antigen. As soon as they are entering our body, our immune system will recognize them, then they will respond and they will remember also. So in case in future there is again these foreign antigen are entering in our body, they are having memory cells and they will remember, and suddenly they will be producing what antibodies to fight with these foreign antigen. The immune system, it plays role in allergic reaction, autoimmune disease, and organ transplantation. The immune system include what are different parts of immune system: lymphoid organs, tissues, cells, and antibodies. So these are some of the part of immune system. Lymphoid organs, tissues, cells, and antibodies, they are a part of immune system. Now we'll be discussing about lymphoid organs. Lymphoid organs. These are the organs where origin maturation, and proliferation. Proliferation means for division. So these are the lymphoid organs are the organs where origin, maturation, and proliferation of lymphocytes occur. Lymphocyte means there are WBC. They are also known as lymphocytes. So lymphoid organs are two types. Lymphoid organs are two types. First is primary lymphoid organ, second is secondary lymphoid organs. So now we'll be discussing what are primary lymphoid organ and what are secondary lymphoid organs. So lymphoid organs are a part of immune system. Primary lymphoid organs. These organs, the organ where lymphocytes are matured and differentiated to antigen-sensitive lymphocyte, that is called primary lymphoid organ. So the organ where lymphocyte, WBCs, they are matured and differentiated. There are different types of WBCs are there. So where the organ from where they are matured and where differentiation of lymphocyte takes place, that organs are called primary lymphoid organ. These two types, bone marrow and thymus. So bone marrow and thymus, they are primary lymphoid organs. Sometimes question will be there. Which of them are this primary lymphoid organs? So bone marrow and thymus. These are what? Primary lymphoid organ. So there are red bone marrow, and this is yellow bone marrow. And this is what? Thymus. We'll be discussing about now bone marrow and then thymus. Now bone marrow, that is primary lymphoid organs. The site of formation of all blood cells, including B lymphocyte and T lymphocyte, that is called bone marrow. So this is a what? Bone. Here there are different types of cells are there. This is a marrow. This is spongy bones. There is a marrow, that cavity is there. This is outer is what? Compact bone. Now here this area is magnified. So here they will be seeing what? Adipocytes, normoblasts, myoplaques, eosinophil, melanocytes, erythrocyte, melanocyte. So these are the area where all blood cells, including B lymphocyte and T lymphocytes, are formed. Then second is thymus. It is a bilobed organ seen near the heart and beneath the breastbone. This is what? Thymus. What is the location? It is found near the heart and beneath the breastbone. This is the breastbone. It is below the breastbone. The size of thymus gland is large during birth, but gradually reduces in size and become very small in puberty. As soon as we attain this adolescence start, the size become very small. Immature T lymphocyte from bone marrow is migrated to thymus and matured here. So here these lymphocytes which are immature, they are migrated to the thymus and here they get what? Matured. Now secondary lymphoid organ. The organ to which mature lymphocyte migrate from primary lymphoid organ interact with antigens and then proliferate to become effector cell. Example, spleen and lymph nodes. Spleen and lymph nodes are the example of secondary lymphoid organ. So primary lymphoid organ example is bone marrow and thymus. Secondary lymphoid organ, there are many more. So spleen, lymph nodes, tonsils, Pierce patches. So these are normal tonsils. Here these are what? Inflamed tonsils. Pierce patches are present in the wall of what? Intestine. These are what? Pierce patches are there. So they are found in the what? The small intestine. Sometimes question will be there. Where you'll find Pierce patches. Pierce patches are which type of lymphoid organ, primary or secondary? What is the location of tonsil? So these are the location of tonsil, and this is what? Pierce patches. Apart from this, there are many more. So mucosa associated lymphoid tissues. They are abbreviated as MALT. So the organ to which mature, these are the what? Mucosa associated lymphoid tissues. They are also what example of secondary lymphoid organ, and then appendix, it is also example of secondary lymphoid organ. So you have to remember these names: spleen, lymph nodes, spleen, lymph nodes, tonsils, Pierce patches, mucosa associated lymphoid tissues, appendix. These are the examples of secondary lymphoid organs. Bone marrow and thymus, that is the example of primary lymphoid organs. Now what is spleen? It is a bean-shaped organ, contains lymphocyte and phagocytes. So here it is present here. It has on this what? Side of the stomach. Removes worn out RBCs. So RBCs which are become non-functional, it is removed by spleen, and microorganisms from blood. It is reservoir of erythrocyte in fetus. In the fetus, reservoir of which is organ is the what? Reservoir of erythrocyte. So the answer will be what? Spleen. Now lymphatic lymph nodes. Secondary lymphoid organ is what? Lymph node. They are found in lymphatic system. They trap microorganisms or other antigen. Trapped antigen activate lymphocytes and cause immune response. So this is what one lymph nodes. This is outer region contain lymphocyte cells that detect and destroy harmful bacteria. Lymphatic vessel, this is a lymphatic vessel. Lymphatic vessel carries lymph into lymph nodes. So this is lymph node. There is an inner region contain macrophages, cells that hunt and eat harmful bacteria. Through these lymphatic vessels, they will be coming inside lymph nodes, and here there will be some macrophages will be there. They will be hunting and they will be eating harmful bacteria. There are walls. These walls always open in only one direction. Then back, so if any blood is coming, blood is containing antigen, bacteria, they will be inputted here in the nodes, and here there are macrophages are there. These macrophages will kill these harmful antigen, they will neutralize harmful antigen, bacteria, and then it will be, then again this, the lymph will flow outside of this. MALT means mucosa associated lymphoid tissues. They are also the example of secondary lymphoid organ. MALT, that is what mucosa associated lymphoid tissue, is located within the lining of respiratory system, digestive system, and urogenital tract. It constitutes 50% of lymphoid tissues. So MALT constitute, what is the percentage of MALT? 50%. Where you will find? They will find in the lining, means layer of the respiratory, digestive, and urogenital tract. So these are what, suppose this is a tract. So here there will be what? Mucosa, membrane lining small intestine. So here there are lymphoid follicles are there. Now I will be discussing about immunity. Once you understood what is the immune system, now immunity. Immunity, what is immunity? Ability of body to fight with these pathogens, microbes, virus, bacteria, to protect body. So that is called immunity. It is the ability, what is immunity? It is the ability of immune system to fight the disease causing organism. So there are different types of immunity. Types of immunity: innate immunity, that is inborn immunity, immunity present since birth, that is called innate immunity. Acquired immunity means also known as adaptive immunity. So this, there are in our body some cells are there like macrophages. As soon as pathogen is entering inside our body, they will eat these pathogens. So it is an example of innate immunity. Production of antibodies, that is a acquired immunity. That is what? Acquired immunity. Now innate immunity. What is innate immunity? It is non-specific, means it work for all type of antigen, whether it is bacteria, virus, fungi, any chemical. So it is what? Not specific. It is not specific defense present at the time of birth. It provides barriers to the entry of foreign agent into our body. Barriers are four types. Physical barrier: before entering inside body, they have to cross one physical barrier, that is skin. So skin is a part of innate immunity. Then physiological barrier: if they are crossing what? Skin. So there are some physiological reactions are taking place inside body. So they cannot survive the enzyme, they cannot survive the acidity. Acid. So that is called physical barrier. Then there are some cells are there, they will eat. Then there is what? Cytokinin barriers. So these are what? Innate immunity is having four different types of barrier. First is what? Physical barrier. Second, physiological barrier. Third, cellular barrier. And fourth one is what? Cytokinin barriers. Now what is physical barrier? Physical barrier is skin, prevent entry of foreign bodies. Then mucous coating of the lining, the respiratory, gastrointestinal and urogenital tract to trap microbes. So this is a normal skin barrier function. This is skin, pathogens are coming. So harmful substance unable to penetrate, they cannot cross this skin. So this is what? Physical barrier. So some mucus is also there, they have crossed, so there is some mucus is there, this mucus coating, then epithelial cells, then tissues are there. So this mucus will kill these harmful bacteria. So these mucous membranes are present in the respiratory system, in the lining of respiratory system, gastrointestinal system, and urinogenital tract. So first line of this work, it work as a first line of defense, like saliva, antibacterial enzyme, it is having tears, active antibacterial enzyme, skin, prevent entry of pathogens, mucus, trap dirts and microbes, gastric acid, low pH kills pathogen, good gut bacteria outcompete bad gut bacteria. So this is what first line of defense, physical barrier. Then physiological barrier. They prevent microbial growth. It includes gastric acid, saliva. Here, this is what? This is a stomach. Here, there is saliva is there. Special type of cells are there called parietal cells. They secrete saliva. So it also, in case bacteria is entering here, it will be killed because of gastric acid. Then saliva is also there. It is also having antibacterial enzyme. Then tears are also having antibacterial enzyme. So they keep our body protected from these pathogens. Then third line is what? Cellular barriers. Cellular barriers. Phagocytes like WBC neutrophils are polymorphonuclear leukocyte (PMNL). Monocyte and natural killer lymphocyte, the macrophages. These provide what? These are cellular level of barriers. So these are neutrophil, these are monocyte, these are natural killer cell, then macrophages. They will be killing the pathogen entering inside our body. So this is a bacteria. This is white blood cells is there. So ultimately it will come and it will engulf this bacteria. Okay, this is a bacteria. These bacteria are engulfed by these macrophages. Then cytokinin barrier. So virus infected cell secrete a cytokinin protein called interferon. What is interferon? It is a what? A protein secreted by virus infected cell, not bacterial infected cell. Virus infected cell secrete a protein, cytokinin protein called interferon. It protects non-infected cell from further viral infection. So this is a virus attacking a bacterial cell, then it will produce one interferon. It is having INF gene, IFN gene. So it will synthesize interferon. This is infected cell. This interferon, it is what? Nearby non-infected cell. Okay. So it will be producing, it will giving signal IFN attached to the receptor and then signal activation of genes. Then it will synthesize antiviral protein. So other bacteria which are produced here, they will not be able to infect this cell. Degrades virus nucleic acid. They will be entering. So as soon as virus is coming and attacking this, it will degrade the nucleic acid. So it blocks the virus replication. So what is interferon? Interferon is a protein which is produced by virus infected cell. It protects non-infected cells from further viral infections. Now acquired immunity. What is acquired immunity? It is a pathogen specific. It will be working against particular pathogen one. It's a poliovirus. So poliovirus is there. So it will be working against poliovirus only. So it will become what? Specific. Pathogen specific immunity developed during lifetime is called acquired immunity. It is not present since birth. It is characterized by memory, that is, during first encounter of a pathogen, body produces primary response in low intensity. Second encounter of the same pathogen causes a secondary, and that secondary response is also known as anamnestic response, and it is a higher intensity. So this is what, this one is first time. B cell is being infected by bacteria. So it will be showing a primary response. Primary response is of low intensity. So it will remember. So second time, if same bacteria is affecting this, coming inside the body, it will producing what? Secondary response. What is the other name of secondary response? Anamnestic response. And anamnestic response is what? In a high intensity. So the if you time concentration of antibody, initial exposure is very less. So this is what primary immune response. Then suppose it is recalled. Again, there is a attack of the same bacteria, it will producing secondary immune response. Secondary immune response is also known as anamnestic response. It is a low intensity. It is a low intensity. Acquired immunity, primary and secondary immune response are carried out by with B lymphocyte, B cells, and T lymphocyte, that is T lymphocytes. So primary and secondary immune response are carried out with B lymphocyte, that is B cell, T lymphocyte, that is T cell. So this is a B lymphocyte, this is what T lymphocyte. B lymphocyte produces antibodies. So this is a B lymphocyte. So B cell, it will be producing antibodies. It will be producing what? As soon as antigen is binding here, there are some helper T cells are there, T lymphocyte, they will be helping this, they will be giving chemical signal. So B lymphocyte become very active, and they then will be producing antibodies, and they will be releasing these antibodies in the blood. So the which cell produce antibodies? B cell or T cell? B cell. These are the proteins to fight with pathogens. So what is antibodies? These are the proteins to fight with pathogen. T lymphocyte help B cell to produce antibodies. Structure of antibody. If we see the structure of antibody, an antibody has 4 polypeptide chain, 2 light chain and 2 heavy chain. That's why it is represented as H2L2. So there are 2 heavy chains. This is heavy chain, this is heavy chain. So there are 2 heavy chains, and this is light chain, and this is also light chain. This is light chain. So 2 light chains and 2 heavy chain. This is what? Heavy chain. So these heavy chains and light chains, they are bonded with, they are connected with each other by disulfide bridges are there. So this is disulfide bridge. This is disulfide bridge. Disulfide bridges are there. In these both, there are 2 regions, constant region and variable region. So this is what? Variable region. This area is what? Variable region on light chain, and this is variable region, that is on heavy chain. So both heavy chain as well as light chain, they are having variable regions as well as constant region. So this is a constant region, and this is also constant region. This is also what? Constant region on heavy chain. So constant region of heavy chain is bigger than the constant region of this light chain. So total number of how many peptides are there? 4. 1, 2, 3, and 4. There are different types of antibodies. Antibodies are of different types. They are named that in Ig means immunoglobulins. Ig means immunoglobulins. So immunoglobulin A, IgA, IgG, IgM, IgE, and IgD. So 3 plus 2, 5 different types of immune antibodies are there. This is Ig immunoglobulin G, this is immunoglobulin E, this is immunoglobulin D, this is M, and this is A. Types of acquired immune response. Acquired immune response are 2 types: humoral or antibody-mediated response or antibody-mediated immunity. It is also known as AMI, antibody-mediated immunity, or humoral response. Second is cell-mediated immunity or cell-mediated immunity, that is CMI. In humoral or antibody-mediated response, body releases B cell releases what? Antibodies. In cell mediated, cell itself, see, these are pathogens are coming. So their cells are having what? Some site where they will attack the pathogen. So cell mediated immunity and antibodies mediated immunity. Humoral or antibody mediated response, antibody mediated immunity. What is this? So it is the immune response mediated by antibody. This is the antibodies code. This is a virus. Antibodies are found in blood plasma, so called as humoral immune response. Why they are known as humoral? Because they are present in blood plasma. So this is what antibodies code. This is a virus. These antibodies are there. So this antibody will surround this virus. So virus cannot infect because of this. There are some spikes are there. With the help of these spikes, this virus will attach itself with the uninfected host cell. So at this spike, this antibody will form bond. Okay. So this virus will not be able to infect the host cell. So virus cannot infect the cell. Then tagged virus is destroyed by what? Neutrophils. This virus is tagged. Now the neutrophil will come and it will eat the virus. So this is what acquired immunity. Cell-mediated response or cell-mediated immunity. The immune response mediated by T lymphocyte or T cells. Cell-mediated is what is CMI? It is the immune response mediated by T lymphocyte, not by B lymphocyte. What T lymphocyte does? The body can be differentiate self and non-self, and the CMI cause graft rejection. So T cell make contact with released granules. There are some granules are being released. The virus infected host cell. This is what host cell, and this host cell is infected with the virus. There are virus particles are there. Major histocompatibility complex, that is a protein displaying viral antigen. Granule-induced cell-to self-destruct virus cannot replicate. So this T cell will release some granules. Because of these granules, these cells will kill itself. Therefore, virus will not be able to replicate. This virus will be able to replicate till the time the cell is alive. When this T cell releases some granules, this cell will kill itself. So if the cell is killed, who will be replicating the DNA of virus? So virus will not be able to replicate. This is the way how CMI functions. Tissue matching, when there is an organ transplantation, tissue matching and blood group matching are essential before undertaking any graft or transplant. After this, the patient has to take immunosuppression all his life. Matching donor with patient. This is a donor, this is a patient. There is a type of special cells. They need to be, a test is there, HLA test. So these will be tested. What is recipient? Blood group is okay. Recipient is O, so it can be what? Blood donor is what? Recipient is O. Okay, blood donor is what? O will be. If you are, it will be organ will be accepted. It is recipient is A, so O and A will be accepted. B, O and B will be accepted. A, B. So in this way, matching is done. Types of acquired immunity. Acquired immunity is two types: natural, artificial. Acquired immunity is two types, active and as well as passive. So active is what natural and artificial, passive is natural as well as artificial. So natural, that is active immunity, that is natural, it is created because of infection, or artificial by vaccination. Passive immunity by natural, the maternal antibodies are monoclonal antibodies. So this is what passive immunity. Passive immunity is not being prepared by body. It is the baby is getting the immunity from mother, and patient is getting antibodies from some other person. So body itself is not designing. Therefore, it is what passive immunity. Active immunity is what when body itself is what producing this developing what ability to fight with the pathogen. It may be by infection or vaccination. Now, what is active immunity? It is the immunity in which antibodies are produced in a host body when the host is exposed to antigen. This is a host. Host is exposed to some antigen, virus, bacteria by infection, leaving our dead microbes or other protein. It is a slow process. It is produced by two ways: natural active immunity, artificial active immunity. Natural active immunity means it is developed during natural infection by microbes. Microbe is infecting the person, so person will develop active immunity, natural active immunity. Second is what? It is developed by injecting microbes because of vaccination. So it is developed by injecting microbes deliberately during immunization. So this is what artificial active immunity. So in this way, we have discussed about what active immunity, that is a natural active immunity and artificial active immunity. Then passive immunity. So now this is acquired immunity, that is a passive immunity. Passive immunity. Here ready-made antibodies are directly given to body. These two types: natural passive immunity, artificial passive immunity. Natural passive immunity, antibodies, immunoglobulin G from mother, placenta, from placenta to fetus. It is produced by placenta, and from placenta it is passed to the baby. So antibodies, immunoglobulin G, immunoglobulin A is present in colostrum. So fetus also get antibodies from the placenta. Which antibody? IgG. And in colostrum, first male secreted by mothers in first few days of delivery, that is having antibodies A. So in this way, it is what natural passive immunity. Second is artificial passive immunity, anti-tetanus serum. A person is getting anti-tetanus serum, that is a artificial passive immunity. Immunization. What is immunization? This is based on memory of the immune system. It is two types: active immunization and passive immunization. What is active immunization? That is also known as vaccination. In this, a preparation of vaccine, antigen, protein of the pathogen or inactivated pathogen is introduced into body. It results in the development of antibodies. So during actual infection, the antibodies neutralize the antigen. So this is what active immunization. The vaccine also generate memory B and T cell. They recognize the pathogen quickly. The vaccine injected also generate memory B cell and T cell. They recognize pathogen quickly. Example, polio vaccine, hepatitis B vaccine, DPT vaccine. These are some of the examples of active immunization. Vaccines are produced using DNA recombinant technology. DNA stands for deoxyribonucleic acid, recombinant technology. Like a hepatitis B vaccine is produced from the yeast. So this is a polio vaccine. This is hepatitis B vaccine. Corona vaccine is also there. DPT vaccines. Now, passive immunization. What happens here? It is the direct injection of preformed antibodies or antitoxin. It is required for quick immune response. Example, immunization against tetanus, snake venom. These are what? Immunization, passive immunization. Now, allergy. What is allergy? It is the exaggerated response of immune system to some antigen seen in the environment. What is allergy? Exaggerated response of immune system to some antigen. These antigens are seen in the environment. What is allergy is caused by allergens. Those materials which causes allergy, they are known as allergens. Substance causing allergy, that is called allergen. Like example, mites in dust, pollen, animal dander, force, and others. Antibodies produced against the allergens are called immunoglobulin E, antibody E. So this is what it is. It fights against allergy. Antibody E binds on mast cell to release chemical like histamine and serotonin from them. It results in allergic reaction. So it releases, in case of this allergy, which chemicals are secreted from mast cell. So example, histamine and serotonin. So this baby is having what allergy from these pollen grains. There is a allergy from tick and mites, dust, pollen grain, mold, pet dandruff. So these are the allergens. They will secrete what? Histamine and lymph and serotonin. So this is what antibodies E. It will bind with these allergens and ultimately it will be neutralized. Symptom, what are the symptoms of allergy? Sneezing, watery eyes, running nose, sneezing, watery eyes, running nose, difficulty in breathing, wheezing, skin rashes, and many more. How to determine the person is suffered from allergy or not? Determination of cause of allergy. The patient is exposed to or injected with very small dose of possible allergen and the reaction is studied. What is the treatment of allergy? A drug like antihistamine, adrenaline, and steroid quickly reduce the symptom of allergy. What is asthma? Is a respiratory disease due to allergy. Asthma, asthmatic patient, they are because of this disease is because of allergy. Modern day lifestyle results lowering of immunity and more sensitivity to allergens. Many children in metro cities suffer from allergies and asthma due to sensitivity to the environment. This is due to protected environment provided early in life. So if you are tendering your children too much, if you are being tendered, there is a problem. You'll become more sensitive. So just go in the playground, play, mix with the people, then you will not suffer with any type of allergy. Then autoimmunity. In higher vertebrate, like human beings are higher vertebrate. Memory-based acquired immunity evolved based on ability to differentiate foreign organism from self-cell. What is foreign and what are the self? Sometimes, due to genetic and other unknown reason, the body attacks self-cell, resulting in damage to body. It is called autoimmune disease. So, this is a normal. Sometimes body start damaging its own body parts. So that is called autoimmunity. One example of this type of disease is called autoimmune disease. Give one example of autoimmune disease. So what will be answer? Rheumatoid arthritis. Rheumatoid arthritis. This is normal finger. This will become a finger of person suffering from rheumatoid arthritis. There is a erosion of bone. There is erosion of bones and displacement of bone. Erosion as well as displacement of bone. So in today's lecture this much. In next video, I will be discussing about what is cancer, what is AIDS. So stay connected with my channel. Thank you very much.